Hi, welcome to this Physionic extract wherein I'd like to briefly discuss an incredibly cool discovery looking at how the circadian rhythms in our body affect our cells.
Learn Your Body, a science-based education.
Now, this particular research comes from a massive paper that I dissected looking at exactly that. So you can check out that paper as well as more detailed notes on the figures that I'll be discussing, the data that I'll be discussing here attached to this content, and is entirely free. So certainly check that out and critique it at your own leisure. So what did this paper exactly find out? Well, they tested intestinal cells. So intestinal cells are made up of a variety of different cells, and that's going to be important as we go forward because there's a difference between non-dividing cells and dividing cells. Non-dividing cells in this particular context are like Paneth cells. Paneth cells are immune cells that essentially protect our intestinal tract. Now, dividing cells, on the other hand, can be stem cells or semi-stem cells called progenitor cells. And I want you to keep that in mind because that's going to be important as we move forward. But those all grouped together create intestinal cells. So what these researchers did is that they removed intestinal cells from mice and then they tracked the expression of a particular circadian rhythm gene called PER. Now, PER will then have an impact on a variety of different physiological outcomes. But in this particular study, they wanted to look at how cells divide. So do they divide based off of the circadian rhythm? Is it essentially controlled by the circadian rhythm? And what they initially find, at least based off this data, is that every 24 hours you see a pulse of this circadian rhythm, the expression of these particular genes. Now, on the other hand, however, when they looked at the mitosis or the cell division that occurs based off of that circadian rhythm, they did find a bit of rhythmicity, but that rhythmicity in terms of the actual cell division itself occurred every 12 hours. So there is a link there, but it's not 24 and not 24. So it's not a 1-to-1 ratio, it's more of a 1-to-2 ratio. But then they found that, of course, there were differences in terms of not all the cells divided exactly at 12 hours. Some of them took a little bit longer, some of them took a little bit less time. So they split the cells up based on their orientation or their placement within the intestinal wall. And they found that cells that were lower in the intestinal wall, so the crypt of the intestine compared to kind of the top section of the intestine, tended to divide much slower than the ones that were higher up. The ones that were higher up tended to divide much faster. So then that showed that there was some level of heterogeneity, that it wasn't exactly all the same. So then that got them even more intrigued. They wanted to find out some more information on how the circadian rhythm could be affecting both of these populations of cells. And that's why I mentioned the differences between progenitors and stem cells and Paneth cells, or non-dividing and dividing cells. And interestingly, what they found is that non-dividing cells do show an expression based off of the circadian rhythm. So they show a connection to the circadian rhythm. However, dividing cells do not, which is really strange because we still have evidence that the dividing cells divide based off the circadian rhythm. But the circadian rhythm does not seem to have a direct impact on these particular cells. So what is going on? What is happening to the non-dividing cells that's then leading to this change or this division that has a certain level of coordination that is found in the dividing cells? And here they find that if they change the environment of the cells, meaning that they take away the normal media, so the media is the liquid that the cells are found in, that keeps them alive. If they remove it and put stem cell media specifically, that the circadian rhythm disappears. However, when they take that off and then they put back on the normal media that feeds all the cells, including the non-dividing cells, suddenly the circadian rhythm comes back. So now they're getting more and more clues that there is a relationship, but it's not directly on the dividing cells. So next what they did is they eliminated some of these non-dividing cells, the Paneth cells specifically. So they ablated them, so they deleted them, they got rid of them. And then they wanted to know what happens to cell division. And what you can see is, if you're looking at the data, that there is a loss in that oscillation. You don't see a decrease in cell division and then a sudden increase in cell division. It's not like you have these peaks and troughs. It's just a continuous rise, but you do still see cell division. So it seems that the circadian that has an impact on these non-dividing cells does still have an impact in the way that these cells divide, in terms of, is it more of a flowing kind of an oscillation back and forth, or are they actually just continuously dividing over time and in kind of in a linear fashion? So this provides evidence in that regard that Paneth cells are important, that non-dividing cells are important in terms of this translation of the circadian rhythm to the dividing cells. And then finally what they did is they completely knocked out the genes for the circadian rhythm, and they wanted to find out, in a population of intestinal cells, including non-dividing cells and dividing cells, what would happen if they knocked out the circadian rhythm genes. And what they find is that you do see a decrease, actually a complete loss of division. So these cells are suddenly highly dependent on the circadian rhythm. So what is going on here? Well, the researchers speculate that what's happening is that you're getting binding of particular factors to the intestinal wall, to these Paneth cells specifically, as well as other non-dividing cells in the intestine, from something like serotonin or some other circadian rhythm agonist, promoter that comes from the brain and from the rest of the body. And that leads to the expression of this PER gene, which is the circadian rhythm gene, which then leads to the expression of a protein called Wnt. WNT. So Wnt is then released from the Paneth cells, from the non-dividing cells, which then bind the dividing cells and leads to their division. However, the effect of Wnt on these cells is different. So there is a difference in terms of the speed of this mitosis, this cell division, based off of the Wnt signaling that is released from these non-dividing cells. So the circadian rhythm does not have a direct impact, based off of this paper at least, on the stem cells and progenitor cells, but it does have an indirect impact by impacting the non-dividing cells, which then communicate to the dividing cells. Some really, really cool research here, and this could certainly have implications in health, in that we can have some control over our circadian rhythm, and that could have an impact in terms of cancer risk, in terms of just our ability to grow new intestinal cells and all kinds of other areas of that nature. Now keep in mind that this is simply looking at intestinal cells. This does not necessarily mean that this translates to all the other different tissues in the body, but this is certainly telling that the circadian rhythm does have a differential impact on the type of cell that you're investigating. So really, really cool research here. So hopefully you found that informative. Of course, you can check out my notes, which I will have in full detail linked to this piece of content, as well as the paper itself with notes within that paper as well. And with that said, I hope I have the absolute pleasure of speaking with you in the very next one. Have a good one, guys. Bye.